students, today's topic is step in sexual life cycle of a typical angiosperm plants. In last video, I discussed types of the reproduction: vegetative reproduction, asexual reproduction and sexual reproductions. Vegetative and asexual reproduction is the simple types reproduction, but sexual reproduction is the typical type or complex type. In sexual reproductions, male and female both organisms are necessary. In this type, reproduction in plants contains flowers. You know that plants are two types. First, angiosperms, second, gymnosperm. Angiosperms are plants which contains well-developed flower, fruiting and seeding. But in gymnosperm plants did not contains flowering, fruiting and seedling. Some steps given there which completes the sexual life cycles of flowering plants. First step of developments that leads to formation of male gametes or sperm. You know that male gametes called sperm and female gametes called ovum. Second step is the development that leads to formation of female gametes. After maturing male and female gametes, next step is the fertilization. After fertilizations, development of embryo and seed formation. These are all steps combined then complete sexual life cycle of any angiosperm or flowering plant. First, I explain first step of male gametes development. In development of male gametes or sperm, first is the development of anther or microsporangium. Anthers are microsporangiums. Anthers are which place that male gametes are present. So this is structure. These are the anther. It is the swelling part attached with filament. These all structures called stamen. It is the male part of the flower. When anther is mature, then pollen grains are developed. Next is the formation of microspore. Microspores called pollen grains, and the development or formation of microspore called microsporogenesis. Microsporogenesis is the method of formation of microspores or pollen grains. Next step of pollination. Pollination are explained in last video. The process of reaching pollen grains in female stigma called pollination. In detail, I explained in last video. Next is the development in microspore to form mature male gametophyte and formation of sperm. Pollen grain are the unmature male gametes. After microspore, development of microspore to form mature male gametophyte called sperm. Next is the development that leads to formation of female gametes or embryo sac formation. Female gametes contain which place called megasporangium. So, formation of female gametes first developed of ovules. Ovules is the female part of the flower. This is called megasporangium. It is the larger part of the flowers. Next step is the formation of megaspores. Megaspores are called egg cell. Egg cell is the single larger cell. This process is called megasporogenesis. You know that when development of microspores called microsporogenesis, when developed of megaspores called megasporogenesis. The next step is the development of megaspores, formation of female gametophytes called embryo sac and female gametes. After the male and female gametes, fertilization is occur. The fusion of male gametes and female gametes process called fertilization. When this fertilization present in internally the body called internal fertilization. Mostly plants have internal fertilization. Next are the development of embryo and seed formation. The last step of sexual reproduction is the development of embryo. After fertilization make zygote, then zygote developed embryo. At last embryos developed seeds. This is the last stage of the sexual reproduction. Now I explain the sexual reproduction steps in detail. Here I explain the first point, development of male gametes or formation of male gametes. So first point is how anther are developed and what is the microsporangium. So this is structure how anthers are tetrad conditions are four chambered. These called microsporangia. The development of microsporangium is the eusporangiate type. Means it is developed in a grouped. In grouped condition called initial cells are eusporangiate types. This is the group of microsporangia. The inner part of the tapetum, it is the most important part of the microsporangia, and outer part called epidermis. One part developed and forms well developed pollen grains or sperms. Tapetum is the inner layer of anther wall. It is usually single layered and rich in reserve food materials which provide the food for developing sporogenous tissues or microspores. Because microspores are the living organism or living cells, so it needs the nutrients, and tapetum provide the microspores nutrients. The cellular structure of one part is here. These are the microspores, mother cell, and it is the grouped condition. Outer wall is the epidermis, inner wall is the endothelium, and it is the middle layer, and rich nutrient parts called tapetum. Next point is the formation of microspore or pollen grains. When microsporangium developed and contains maturity, then starting formation of microspores or pollen grains. This process is called microsporogenesis. These microspores or mother cells are 2n. 2n means haploid. So this chart, microspores mother cells are 2n or 2x. It is the diploid cells. And first meiosis division, each cell divides in meiotically or by meiosis to form two haploid cells. Two haploid cells means N or XX cells, or two haploid daughter cells. In second meiotic second division, it forms 4 microspores or pollen grains. This called M1 phase or meiotic first division, and this called M2 and meiotic second division. Firstly, it divide 2 haploid or 2 daughter cell. These cell called microspores or pollen grains. Then secondary, it divides and developed 4 microscopes or pollen grains. It is the haploid cells. So pollen grains, the microscopes or pollen grains are formed, remain associated with tetrad condition. This group of 4 microscopes or pollen grains is called microscope tetrad. Different plants represent 5 type tetrads or condition of microspore or pollen grains are 5 types. First, tetrahedral tetrad means 4 groups contained in 4 groups. It is the most common containing stage of the pollen grains. Next is the isobilateral tetrad. Next is the textate tetrad and inverted T-shaped tetrad. Last is the linear tetrad, but mostly plant present tetrahedral tetrad. So this structure tetrahedral tetrad contains 3 types. Isobilateral tetrad means 4 pollen grains are contains opposite side. These are the discrete tetrads. These are the 1, 2, 4. 4 pollen grains are contained here. These are the inverted T-shaped tetrad. And last is the linear tetrad. Linear and T-shaped tetrad is rare in case, but sometimes plants contains 4 types tetrad also. Here we show the structure of one microspore or one pollen grains. It is unicellular cell. Uninucleated means single nucleus is present. Haploid means single N. Spherical structure and developed rise to one male gametophyte. One microspore represented one male gametophyte. It surrounded two-layered wall, first and second. Outer layer is called exine and inner layer is called intine. Outer layer is called also epidermis. Inner layer is called intine. These are the surrounding by nuclear part. Exine shows different types of outgrowths. It is not similar layer. So this structure, it contains many outgrowths. Intine layer is smooth and cellulosic, but exine layer are rough and fatty substance. The under the intine layer contains a liquid material called cytoplasm and some porous-like structures called germ pores. In exine layer present some porous type structure. So here these called germ pore, and one nucleus are present called haplide nucleus. So these are the structure of microspore. One importance of germ pore is because these holes or these marks are origin of pollen tube in the fertilization period. When male gametes fertilized in female gametes in the ovule, germ pore germinate a long pollen tube where haplide nucleus are fused to female gametes. So it is importance of microspores. In difference between monocot and dicot plants, microscopic. In dicot plants, 3 germ pores are present. In pollen grains or microspores of dicot plant contains 3 germ pores, but in monocot only 1 germ pores are present. If microspores present 1 germ pore called monocolpate, when 2 germ pores are present called bicolpate, and when 3 germ pores are present, pollen is called tricolpate microspores. So dicot plants are tricolpate microspore. The next step is development of microspore. After formation of microspore, starting development of microspore are matures of microspores and formation of male gametophyte. When developed or mature microspores then form male gametophyte or sperm, or mature microspore called sperm or male gametes. Development of male gametophyte start when a pollen are within anther lobes. These are the anther lobes. When pollen grains present in anther lobes starting the maturing stage. Some part of development complete microspores in microsporangia or anther lobes who contains 2 or 3 cell stage gets completed, then half development of microspore complete after pollination. When male gametophyte take place on a stigma. In development of male gametophyte involves following steps. First step is development of male gametophyte before pollination. Before pollination development I explained in first and descent of anther and liberation of developing pollens. This condition, descent of pollen grains or microspores in anthers. So these are the condition of descent of anther and liberation of developing pollens. When liberate the developed pollens in female stigma, then start pollination. When pollens are reaches in female parts stigma, these process called pollination. After pollination, when pollen tubes germinate in germ pods and male gametophyte reach this pathway, ovules, then development of male gametes start. Here we show first part, development of male gametophyte before pollination. Development of male gametophyte before pollination occurs inside the anther or microsporangia. During germination, the nucleus of microspore is displaced from center of one side. This displacement is always a particular direction and mark the position of germinative cell or germinative pores. In last condition, microspores developed 3 celled stage, 1 or vegetative cell and 2 or sperm cells. Before pollination, germinate male gametophyte, then start liberation of developing pollens. When liberates the developed pollens in mature female stigma, then start development of male gametophyte after pollination. It involves following steps. First is the formation of male gametes or sperms. So this microscopes of pollen grains, it developed after pollination when it reaches the stigma of the female flower. Only a right kind of spore germinate on the right kind of stigma. This is the germ pores. Formation of or liberation of pollens germinate two cells. First are the vegetative cells and two are the germinative cells. Total are the three-celled structures of pollen grains. Germinative cells called two-celled male gametophyte. Germinative cell present large amount of protoplasm and acquires a characteristics vermiform appearance. It now divides mitotically and to form two elongated haploid male gametes or sperm. So, germinative cells developed male gametes or sperms. These male gametes are genetically similar and remain in paired condition. Because these male gametes developed one type germinative cell, so its are genetically similar and remain in pairs for a period. Next is the formation of pollen tubes. In the place of germ pores, one identical pollen tubes are formate and growths. The axine of the outer wall, these are the axine cell or outer wall also. The pollen now ruptured and the cytoplasm of the vegetative cells through germ pores in the form of a tube called pollen tube. Apex of the pollen tubes contains nucleus or two male gametes or sperms. The tubes grow towards the ovules or female parts. The pathway of stigma or styles. So this structure, sperms are here, then pollen tubes germinate, the pathway of style enter the pollen tube and reaches the ovules which contains under the ovary. Usually a single pollen tubes are arises in one pollen grains. Either germ pores are three stages, but a single pores are arises from one pollens. Such pollen grains called monosiphonous. In some plants, pollen grains give rise to many pollen tubes. Such pollens are called polysiphonous. Polysiphonous plants are rare in case, but in most plants are monocephalous. After germination, the pollen tubes, male gametes and vegetative nucleus contains through the style or corpel and towards the mature ovules or reaches toward mature ovules. Here I stopped the development of male gametophyte or pollen grains or sperms. After development of pollen grains or male gametophyte or sperms, then start development of female gametes or egg cells. Pollen grains are very tiny structures or tiny living cells, so it's called microspores, and this process is called microsporogenesis. But females gametes are large in size, this is called egg cell, and these called megaspores. In sexual reproductions, pollen grains are many numbers developed, but egg cells are single number developed. After development of male gametophyte form a microspores. Mature male gametophyte is three-celled. You know that three-celled are two are the male gametes and one are the vegetative cell. In next video, I explained development of female gametes or egg cells, then after fertilization, and last embryo development, then complete the sexual reproduction in flowering plants.